Okay, so today is a big lecture. It's going to be about the cell, and cells are one of the main things that we talk about. Your body is based on cells, on having cells around. The cell is the main unit of life in your body. In fact, the cell is the main unit of life in general. All of the things that your body does are based on what its cells do and how it does them. So in order to understand the biology of the body, which is what anatomy and physiology is, you need to understand the biology of the cells. First I want to talk about cell shapes. All right? Cells come in many, many different varieties inside of your body. You have, potentially hundreds of different cell types and thousands of different variants on those types. But there are a few common denominators. Cells come in different shapes and sizes. This is by no means a comprehensive list, but this is some of the more common ones that you'll see. We will talk about these a lot more when we talk about tissues, but you need to know basically what we're talking about when we talk about cell shapes. First off, squamous cells. Squamous cells, when you hear squamous, you should think squat. So squaw for squamous, squaw for squat. Means short, basically. These cells look, you're actually seeing them from the side view here. They kind of look like fried eggs. They're sort of roundish, mostly flat, with a little raised area in the middle, which is where the nucleus is. They're found mostly in epithelial tissue, which forms the linings of things. You have a lot of squamous cells in your skin. Next, we have cuboidal. As you might guess from the name, cuboidal are cube-shaped. Now, cells are very seldomly actually perfectly cubed shapes. Nature doesn't really like right angles, so they're usually going to have like rounded edges. But basically, cuboidal cells are going to be about as tall as they are wide, approximately. It's never perfect. But if you have cells that are about the same width that they are height, and they have relatively straight sides, maybe some rounded corners, and it might not always be four sides, but that's a good indication of cuboidal cells. And we have columnar cells, as you might guess from the name, column. These are cells that are shaped like columns, meaning that they are taller than they are wide. Columnar cells are usually 2 to 3 times as tall as they are wide, though it can vary depending upon the cell type that you are looking at. All 3 of these top types are commonly found in epithelial tissue, which is tissue that forms the lining of things inside of your body. Polygonal cells are similar to cuboidal cells, but have more than 4 sides. They could have 5, 6, 7, or 8 sides. Once you get more than 8 sides, you can't really tell in a cell how many sides that it has, and it's probably starting to look a little bit more spheroidal. On that note, spheroidal cells, they're round. They're spherish. A lot of your blood cells, your white blood cells in particular, are spheroidal, as are a number of other different types of cells. Cells can be stellate. Stellate means star-shaped, and these have, like, if you think of like an asterisk type star, these are cells that have a round-ish center body, and then projections shooting out in a bunch of different directions. There are a number of different stellate types of cells, but the probably most well-known are neurons, nerve cells. There is another type of cell called a dendritic cell, which is actually an immune system cell, which is also stellate. It puts out these projections that it uses to catch invading bacterial or whatever types of cells. Discoid. This is a fairly rare cell shape type that makes cells that look basically like toruses, these like flattened discs with indentations. The most common and pretty much the main discoid cell that you're going to see in the human body are red blood cells. When we talk about blood, we'll talk about why blood has this shape, but basically it's a way of maximizing the surface area of the cell and minimizing the distance to any particular area, because your red blood cells have to carry as much hemoglobin as possible, but that hemoglobin has to be close to the surface so that the oxygen can be absorbed and released. Fusiform cells are cells that are shaped kind of like toothpicks. They are long with usually tapered ends like this. The most well-known fusiform shaped cell is the smooth muscle cell, but there are a number of fusiform shaped cells in the body. And last we have filamentous cells. Filamentous cells are really long, really long cells, and usually you have multiple cells that have kind of glommed together in a big line. So this is actually a skeletal muscle fiber cell, and it has multiple nuclei in it, and it's actually made out of the fusion of several different types of cells. So these can actually be really long, much longer than you would expect a single cell to be. And they're sometimes referred to as fibers rather than cells, because they have some differences between them and traditional cells. Like I said, these are by no means all of the cell shapes out there, but these are some of the main ones that you might see. So that's what cells are shaped like. What are cells made out of? The function of a cell is related to its form, just like it is with everything else. And just like your body has within it different organs, you've got your brain, your heart, your liver, your kidneys, and these all do different things. They all have their own specialized tasks. Your cells, which are called eukaryotic cells, eukaryotic eukaryotic cells, which is you and every other animal and all of the plants and all of the fungi out there, eukaryotic cells all have specialized what are called organelles inside of them, which are walled off areas that have specialized tasks. This is in contrast to prokaryotic cells, which are mostly bacterial cells, which do not have these specialized compartments. And if you go on to take microbiology, we will talk extensively about prokaryotic cells and bacteria in there. But since we're mostly talking about the human body here, we're going to focus on eukaryotic cells. There are basically three main areas of the cell. You can have, well, inside the cell and outside the cell, and the boundary between those two things, right? That's the basic distinction, is you've got the inside, the outside, and the thing that is the boundary. Well, the inside of the cell, specifically actually the part that is between the cell barrier and the nucleus, which we'll talk more about in just a second, this region is called the cytoplasm. The cytoplasm contains all of the organelles. So like this is an organelle here, this is a big organelle here, here are some organelles. All of these organelles are floating in the cytoplasm. It has inclusion bodies. Inclusion bodies are like non-membrane bound storage or waste areas. It's basically like a big pile of stuff that's all piled and stuck together. And the cytosol, which is the fluid that inhabits the cytoplasm, right? So the cytoplasm contains all of these things, the organelles, the inclusions, everything like that, and the fluid that they're all floating in is the cytosol. A lot of people think, you know, because when we draw it, we always draw the cytosol as being clear, and it sort of looks like these are just floating in this pool of liquid. We think of the cytosol as being like water, but that's actually not true. The cytosol is about 50% water and about 50% other stuff, like proteins, ions, sugars, small molecules, things like that. It's got about the consistency of a soft jello. So it's jam-packed with things inside of it and stuff going on. It's not just like a pool of water that things are floating in. There's organization there. Then we have the boundary, what separates the inside from the outside. And for cells, that is the plasma membrane. We're going to talk a lot more extensively about the plasma membrane in a future lecture. But basically what you need to know is that it's made of proteins and lipids, special lipids called phospholipids, and it defines the boundary of a cell. And the plasma membrane, its inside nature is hydrophobic. That's what makes it a boundary, is that it's got this hydrophobic layer, this lipid layer, and water does not want to cross that layer very easily. So the water from the inside doesn't just escape out, and the water from outside doesn't just escape in, or doesn't just come in. That's what makes it a good boundary. And then outside of the cell, we have what's called the extracellular fluid. Now, if you're like an amoeba or something, and you're just like living in a lake, then your extracellular fluid is the lake. But you are not a single-celled organism. Your body has like 100 trillion cells in it. And the stuff that is in between those cells is just as important for holding your body together as the cells themselves. This extracellular fluid is how cells communicate with each other. It is how cells hold on to one another. It is this area where proteins are passing through, and where a lot of organization takes place, and a lot of exchange takes place. In tissues, which is pretty much most of your body, all of your body that isn't the blood, this fluid is called interstitial fluid. That means fluid in between cells. So this is a model of a cell. There's no cell in your body that is actually going to look like this. This is sort of a cell with all the fixins, right? Everything that could be in an animal cell is in this cell here. But in reality, there are very, very few cells that have everything that a cell could possibly have. And as far as I know, no cells in your body are going to have all of these features. But these are all of the possible features, and some cells are going to have some of these features, and some cells won't have some of these features. For instance, red blood cells don't have a nucleus, and they don't have mitochondria. The only cell with a flagella in your body, in some of your bodies at least, is sperm cells. We have microvilli. These are found in some cells, but not others. And I'm going to explain basically what each of these components does. And that's most of what I'm going to do right now. So we're going to start off by talking about the nucleus. All right? So the nucleus is the control center of the cell. It is where all of the cell's information storage takes place. It's where the cell stores its DNA in the form of genes. Okay. All eukaryotic cells have a nucleus for at least part of their life. Most cells have one nucleus, just like you've got one brain. But there are some cells that actually have more than one nucleus, particularly those filamentous cells that we talked about, where you have really, really, really long cells, and you might have multiple nuclei multiple nuclei going down this cell, because the cell's so long that some parts of it might be too far away from one nucleus in order to get signals from it. So you have, just like you might have heard that like there are some dinosaurs that were so big that they had to have an extra brain in their butt to control the back end. Well, there are some cells that are so big that they have to have more than one nucleus, but most cells have only one nucleus. There are some cells that are anuclear as well. That means that they do not have a nucleus. This is mostly red blood cells. And red blood cells don't start off without a nucleus. When they're born, they have a nucleus, but that nucleus is removed from them before they are dumped into the blood. And as a consequence, once the nucleus is gone, they can no longer reproduce. So all cells start off with a nucleus. Some of them lose that nucleus along the way. The nucleus itself is separated from the rest of the cell. In fact, most organelles, most of these functional compartments, are separated from the cell cytoplasm by a membrane. It's a very similar membrane to the cell membrane that separates the inside of the cell from the outside of the cell. It is again made out of proteins and lipids. The nucleus is separated by a special membrane. It is a double membrane. It's 2 layers thick, and it is filled with what are called nuclear pores. As you might guess, the nucleus, because that's where the brains of the cell are, that's where all of the information is stored, but all of the things are done outside of the nucleus. There's a lot of things that are going into and out of the nucleus all the time. It's like, think of it as like a major office building or a downtown area in a city, right? You've always got lots of things going in, lots of things coming out, and so you need to have very well-defined ways for things to get in and out, right? If you have a downtown area in a city that doesn't have any roads leading in, doesn't have any highways leading in, it's all going to be a huge mess. So your nuclear envelope, the membrane of the nucleus, has these pores, and those pores are going to be what regulates traffic into and out of the nucleus. The nuclear envelope is shaped and held in place. It's given some structure by what's called the nuclear lamina. This is a web of proteins that basically keep the nuclear envelope from collapsing in on itself. And they provide anchor sites for the cell's DNA or chromatin. So when we talk about chromatin is DNA plus protein. And And your DNA is never, in your cells, found just by itself. It's always complexed with these proteins that are going to organize it and regulate it. So this DNA protein complex, this chromatin, is going to be attached to the nuclear envelope through the lamina. Just like the inside of the cell was called the cytoplasm. Cyto, by the way, cyto means cell. And so anytime you see cyto, just think cell. Cytoplasm, a plasm is sort of a gel, means cell gel or cell goo, right? So that's what's inside the cell. What do we call what's inside the nucleus? It's a big organelle. It's probably in most cells the biggest organelle. And the inside of it has got a lot of fluid with a lot of this DNA and protein floating around inside of it, and we call that the nucleoplasm, which means the goo inside of the nucleus. And the nucleoplasm consists of basically two areas. One is the chromatin. And remember, the chromatin is all of your DNA and all of the proteins that are attached to it. Chroma means colored, and it's called chromatin because when you stain the cell, it stains very, very deeply, and it's easy to see. And there is also kind of like a nucleus inside of the nucleus called the nucleoli, which just means little nucleus. Most of the nucleus is devoted to making or storing DNA, but this small area, the nucleoli, is where ribosomes are made. And we will talk more about ribosomes in just a little bit, but you need to know that the nucleoli is where ribosomes are born. So here's a picture of the nucleus. You can see, so this is a surface picture of the nucleus, and the surface, the membrane, is visible here, and you can see all these little dots in it. Those are the pores. This is a electron microscopy picture of a cut through the nucleus. And you can see the nucleolus, which is this dense, small, dense area inside the nucleus, and the nucleoplasm, which mostly contains chromatin, and then you have your nuclear envelope on the outside. So the next thing that we're going to talk about is the endoplasmic reticulum, right? Now, we're starting from the nucleus, which is right here in the center, and we're moving outward from the nucleus. So the endoplasmic reticulum is located right around the nucleus. It's peripheral to the nucleus, and it's actually connected to the nucleus. So you can see, for instance, here and over here and right there. Those are areas where the endoplasmic reticulum, which is this kind of foldy-looking area out here, as well as this sort of tubey-looking area over here. Those are areas where those connect in to the nucleus. So the membrane is actually made from the outer membrane of the nuclear envelope. The endoplasmic reticulum, so endo means inside, plasmic means goo. Basically what that means is that it's the portion of the goo that is located closest to the nucleus, and reticulum, anytime you see reticu, that means net like. net-like. All right? So it's a network, a bunch of areas that are kind of like where you've got different connections between them. There are two connected but completely separate portions of the endoplasmic reticulum. It's like they connect in with each other, they're joined, but they do totally different things. The first is what is called the rough endoplasmic reticulum, which you can see here, right? These things that look like a bunch of sort of flat folds. That is the rough endoplasmic reticulum, and it's called the rough endoplasmic reticulum because if you take a really close look, like with this electron microscopy image right here, you can see all of these little dots that are lining this fold. Those little dots are ribosomes, and they make the rough endoplasmic reticulum have a rough appearance to it, a little studded or dotted appearance. So you find ribosomes in contact with the rough ER. We're going to call the endoplasmic reticulum the ER from now on. The function of the rough ER is that this is where many proteins are made. The ribosomes, the job of the ribosomes is to make proteins. And proteins can be made in one of two places. Right? Some ribosomes just float around in the cytosol, and they just make proteins that then float around in the cytosol. So any protein whose job is to just float around in the cytosol gets just made there. But many proteins are made with a specific destination in mind. They need to go to another organelle. They aren't just going to be floating around. Maybe they go into the nucleus, maybe they go into the lysosome, maybe they actually get shipped out of the cell itself. All of those proteins that have a specific destination, those are all made in the rough ER. Now, why this shape? This is something that is going to come up over and over and over again in this course. And so you should get used to seeing it now. Anytime you have something that looks really foldy or that has lots of stacks or is made out of lots of tubes or little finger-like projections, and you ask yourself, why is it shaped like that? Remember that biology is all about form and function, right? The way things are shaped, they're shaped like that because that is the shape that is necessary for their function. So what function is going to be given by this shape? What does this shape do for it? It increases surface area. Anytime you see one of these motifs where you have lots of folds or lots of fingery projections or lots of tubes and tunnels, you should think surface area. Something is going on that surface that is important. Here, what's going on is that the ribosomes bind to the surface. That's why it's the rough ER. So that rough ER, the ribosomes can only bind to the surface. The more surface you have, the more proteins you can make faster. More surface area, more ribosomes binding, more proteins made. These proteins, as they are made, actually migrate through this set of like interconnected stacks until they get to the outside, and then the proteins all kind of gather inside one portion of a stack, and that portion will break off into a little bubble called a vesicle. And then that bubble takes those proteins somewhere else. We're going to talk about that in just a second. We've got a whole other part of the endoplasmic reticulum. That's the smooth ER. It's called the smooth ER because it does not have proteins bound to it. or have ribosomes bound to it. And the smooth ER, as opposed to looking like stacks, tends to look like a bunch of tunnels or tubes together. So you can see here you've got a bunch of flaps, a bunch of folds. This area here, which is the smooth ER, what you see is much more caverns and circles. Whenever a cavern like goes into or out of the slide, it makes a circle. When it like runs alongside the slide, you see like this tube, and you'll note that you don't see any of those rough spots, no ribosomes associated with it. We know ribosomes make protein. Do you think that the smooth ER is going to be involved in protein production? No, because it doesn't have any ribosomes, so it doesn't make any proteins. These tubey-looking things are called cisternae, and this is actually where lipids are made. This is where steroids and other lipids are synthesized. So all of the membrane in the cell is actually made here in the ER. And then it breaks off from the ER, or it migrates over to the rough ER, and that membrane can then form or be transported to wherever it needs to go. So membrane is made in the smooth ER. Steroids are made in the smooth ER. That's where cholesterol and certain hormones are made. And the smooth ER has a second function. It is where detoxification happens, or it's actually one of two areas where detoxification happens, where toxic chemicals like, say, alcohol and other drugs are broken down. So what types of cells do you think would have lots of rough endoplasmic reticulum versus lots of smooth endoplasmic reticulum? Well, let's say that I have a cell whose job is to make lots of proteins. What's it going to have a lot of? It's going to have a lot of rough endoplasmic reticulum. So, for instance, plasma cells, which are cells that make antibodies, and antibodies are proteins, they have to make a lot of antibodies, so they're going to have a lot of rough ER. What about liver cells? What do you know about the liver? Well, the liver is involved in detoxification. So what do you think that the liver is going to have a lot of? Well, the liver is going to have a lot of smooth ER. There is one specialized function of the smooth ER that I wanted to point out. This is something which is pretty exclusive to muscle cells. But the smooth ER is also used for storing calcium. Calcium can actually screw up a lot of things inside of the cell, but there are certain things that you need it for. And so inside of the smooth ER is a fairly high calcium environment, and then you can release that calcium into the rest of the cell. In most cells, this is a pretty small function of it, but your muscles, the trigger for your muscles to contract is when calcium is released into them. So your muscles have a huge network of smooth ER whose job is really specialized for calcium storage. It still does this detox and making membranes thing. But the reason why they have a lot of smooth, why muscle cells have a lot of smooth ER is to hold this calcium. And when smooth ER is specialized for holding calcium, we call it the sarcoplasmic reticulum. So if you see sarcoplasmic reticulum, that means smooth ER that is specialized in holding calcium. So we see the ribosomes are bound to the rough ER. Ribosomes are not membrane bound. All of these other things that we talked about, so the nucleus has a membrane that surrounds it and acts as a barrier. The smooth and rough ER, these folds are made out of membranes, but the ribosome is not membrane bound. It is often considered to be an organelle, but it's just floating there, non-membrane bound. And ribosomes can be found either floating free in the cytoplasm, or they can be found bound to the rough ER. And wherever they are, that's where they make proteins. So free ribosomes make proteins that go into the cytosol. Bound ribosomes make proteins that go into the endoplasmic reticulum and will eventually be shipped off to other places. Ribosomes' job is to make proteins, and ribosomes are made out of two subunits, what's called the large subunit and the small subunit. All right, so moving outwards from the endoplasmic reticulum, we are next going to talk about the Golgi apparatus. So you can see here, this is the endoplasmic reticulum. You move outwards from that, and you get to the Golgi. That's this green thing here that looks like a stack of pancakes, kind of. It's a stack of long ellipsoid flaps. All right? So when you think, well, why is it shaped like that? What do you think it has to do with? Probably surface area. So the Golgi apparatus has two sides to it. The cis side, cis means same or near, right? So the cis side is the side that faces the endoplasmic reticulum. And then it has the trans side. Trans means opposite side or other side. This side faces away from the nucleus and towards the rest of the cell. The Golgi is where all of the proteins from the endoplasmic reticulum are going to be shipped to. So you have your proteins made in the endoplasmic reticulum. They break off into these vesicles. And the vesicles then go to the Golgi, and they merge with the Golgi on the cis side and dump all of their proteins in there. Now, some of you have probably worked in retail at some point in your life. So you can think of the ER as like the factory where all of this stuff gets made. But it just gets like random boxes. Like it just gets stuffed into random boxes and then shipped to the Golgi. So imagine you've got this huge factory, it makes hundreds of different things. When things get made, they just get put into a box and then shipped to this center. And the Golgi is sometimes referred to as the central shipping and receiving center. So the receiving side gets these random boxes that are just full of a bunch of stuff. You could have, shoes next to clothing, next to baseballs, next to toys, next to furniture. Who knows? Whole bunch of stuff just arriving in random boxes from the manufacturer. And the job of the Golgi is to sort through everything. Go, all right, these things, they are all baseballs. We're going to put them all into one box. That's the baseball box. And then we're going to ship them to sporting goods, right? These are all clothing. We're going to put all of the men's clothing together in one big box, and then we're going to ship that to the menswear. And so what it does is it sorts through the proteins, puts similar proteins that have a similar destination together, organizes them, and labels them. Because all of this stuff arrives unlabeled, right? So you got to slap a barcode on it. Everything gets a barcode. It tells you what it is and where it goes. So these proteins come into the cis side, and as they negotiate their way through this stack of flaps here, this stack of pancakes here, they get labeled, right? The Golgi figures out what type of protein it is, where it needs to go, and then it puts a little label on it. This label is actually made out of carbohydrates. It's a sugar tag, but it acts like a barcode. And then things with similar barcodes that are headed to similar places will all get packed together on the trans side and break off into a vesicle. But now they've been organized. So everything in this vesicle might be headed to a lysosome. Everything in this vesicle over here might be headed to the surface of the cell, or it's eventually going to be headed outside of the cell. And so that's what happens in the Golgi. Things get organized, labeled, packed together, and then shipped off to their final destination. The next organelle, like this is one of the destination points, is the lysosome, right? Lysosomes look kind of like large vesicles. They aren't huge, flappy stacks. They're just these sort of spherical, membrane-bound areas that are kind of floating about a little bit randomly, haphazardly inside of the cell. But what do the lysosomes do? Well, if you remember when we talked about hydrolysis, hydro, water, lysis, to cut, right? Lysosomes are bodies, so some means body or container, and lyse means to cut. So this is going to be a container where things get cut up, where things get taken apart, sliced and diced. This lysosome is actually a body where primarily hydrolysis takes place. So inside the lysosome, you have a whole bunch of enzymes that are hydrolytic, digesting enzymes. This is where proteins get digested into amino acids. This is where nucleic acids get digested into nucleotides, where complex carbohydrates go to get turned into simple sugars, where phospholipids and other lipids go to be taken apart into fatty acids. So it is a digestive organelle. You can think of it as the stomach of the cell. Whenever something needs to be digested, it is sent to the lysosome. What is that likely to be? Well, there are a few different things. One is autophagy, right? Auto means self. So that means self. And phagy means mouth or eat. So autophagy means self-eating. This is where a cell wants to digest part of itself. Why would it want to do that? Maybe it has too much of something. Maybe a cell has too many mitochondria. It doesn't need that many mitochondria. Well, why keep them around then? They're taking up space, they're taking up resources. You could be using the things that it's made out of to make other things. So you label a mitochondria for destruction, and then it combines with a lysosome, and the enzymes of the lysosome will then go inside of it and then digest the whole thing. And it will digest that mitochondria, and then you free up all of those resources to be reused. We're taking our Lego and we are tearing it all apart into component pieces that can then be reused. But that is only a part of the cell. So you're digesting an organelle, not the entire cell. When you want to digest the entire cell, when a cell wants to digest itself, that is called autolysis or self-cutting. This is a form of cell suicide. The cell doesn't survive this process, but your body is not a democracy. Your body is a dictatorship, and if your body decides that a cell needs to die, that cell says, yes, sir, and it dies. And it dies in the most friendly way that it can. It actually starts to digest itself. And it basically just releases its lysosomes. The lysosomes will then digest everything that is inside of the cell, and the cell dies. But while it dies, it like packs everything up all neat and together into super tidy compartments that allow other cells to come by and then reuse all of its materials. It's like a super green, sustainable way for your body to get rid of cells. All right? So lysosomes can be involved in cell suicide or autolysis. They can also be involved in phagocytosis. Remember, phago or phagy means mouth or to eat. Cyto means cell. This is when a cell eats another cell or eats another thing. This is cell eating. Basically, you have one cell, all right, that is going to surround and eat the second cell, which is then inside of it, and then it will digest that cell. Whatever it is eaten will be combined or sent to the lysosome, and then the lysosome digests it. So like when your white blood cells eat a bacteria, that bacteria is then sent to a lysosome, the lysosome digests it, and it eats that. So the next organelle is the peroxisome. Peroxisomes look, under a microscope, almost exactly like lysosomes. They're basically little, they're kind of like big vesicles or little spherical things inside of the cell. But they have a very different function than lysosomes. Specifically peroxisomes, right? You've probably heard of peroxide. Maybe you bought some peroxide from the store, and your mom maybe put it on cuts when you were a kid and you fell off of your bike. Peroxide is used to clean wounds because it's really extremely destructive. It tears things apart. Peroxide is highly oxidizing, and it just rips things apart and destroys chemical bonds. So you put it on some bacteria, and it rips the bacteria apart. You've got dead bacteria. That's why it's used to clean wounds. It kills off all of the bacteria that are there, or at least that's the idea. It actually doesn't work all that well for reasons that we'll get into probably in microbiology. But we can talk a little bit about it here. Your cells make peroxide. When you breathe oxygen, and your body uses oxygen to make energy, one of the byproducts of that is peroxide. And if that peroxide was allowed to build up inside of your cells, they would rip apart your cells from the inside. So instead, you have a defense mechanism. And that defense mechanism is the peroxisome. Peroxisomes contain an enzyme called catalase. Catalase is really, really good at taking hydrogen peroxide and turning it into water and oxygen. So, and water and oxygen, of course, are pretty reasonably safe. So the organelle in your body that uses oxygen and makes energy is the mitochondria. We'll talk about that in just a second. You will often find peroxisomes close by to the mitochondria to scavenge up the peroxides that they make, and then get rid of those peroxides. So they absorb the peroxides and then they neutralize the peroxides. The dangerous thing about peroxides are these chemicals called free radicals. And the peroxisomes have another function, a related function as well, There are some things, some particular bonds that the lysosome doesn't do very good at getting rid of, right? So the lysosome mostly does hydrolysis. There are some bonds that can't be hydrolyzed, and that the smooth ER can't really handle either, but those bonds can be broken by hydrogen peroxide. So since you've got all of this hydrogen peroxide in one place, you might as well use it to break some bonds. And so other things that you want to be destroyed can be sent to the peroxisome. And so the peroxisome can detox things. It's involved in the detoxification of alcohol, detoxification of other drugs, and it also is involved in breaking down fatty acids. Because fatty acids are actually very stable, very difficult to break down, but the peroxisome can do it. So what types of cells are you going to see lots of peroxisomes in? You're going to see them in cells that have a lot of mitochondria, because those have a lot of peroxide to neutralize. And you're going to see them in cells that are involved in primarily detoxification. So you find peroxisomes in pretty much all cells, because all cells have to deal with this oxygen damage, but you find a lot of them in the liver and the kidney, because the liver and the kidney are involved in detoxification. Moving out from the peroxisome, we're going to talk about the mitochondria. Mitochondria is a really cool organelle. There's a lot of really important stuff that goes on here. We're going to talk about it more when we talk about metabolism, but to just give you the overview. The mitochondria is the powerhouse of the cell. It is where your cell's ATP gets made. This is where you take sugar, combine that sugar with oxygen, and extract energy from it. So you use oxygen. You know, you need oxygen to burn things. Things don't burn without oxygen. Your body is burning sugar, and it requires oxygen to burn that sugar, and that burning takes place inside the mitochondria. And the energy that is released from that burning is captured in the form of ATP. So the mitochondria is where sugar and other things, proteins, lipids, stuff like that, are turned into ATP. Now, you see this picture of the mitochondria here? What do you see inside of it? You see all of these little squiggly bits. Those are membranes. That's the inner membrane. The mitochondria has two membranes, an outer and an inner membrane. That inner membrane is all squiggled back and forth. Why do you think that is? It increases surface area. That tells you that there's something important going on that surface. And in fact, on that surface is going to be where ATP is made. The mitochondria can be anywhere from being like these sort of short bean-shaped things to some mitochondria that are actually really long thread-like things that have grow into these long filaments. You'll particularly find those, for instance, in like muscle cells, which of course need a lot of energy. These folds here on the inner membrane are called cristae, which just means creases, and in between those creases is an area called the matrix, which is inside the inner membrane. The matrix is going to contain ribosomes, enzymes that are used for breaking down sugar, and the cristae contains enzymes that are going to take those sugar breakdown products and turn them into ATP. One of the things that's really cool about the mitochondria, so remember how we said that the cell has the nucleus and all of the cell's DNA is in the nucleus, and there are ribosomes that are going to take RNA that is copied from DNA coming out of the nucleus and turn those things into proteins. Well, the mitochondria is sort of an exception to that rule. The mitochondria has its own DNA. mitochondrial DNA. And it makes its own proteins using its own ribosomes. So it acts kind of like a little cell. It has this double membrane, and it has its own DNA inside of here, and then it takes that DNA and it uses its own ribosomes to make its own proteins. Kind of like a little cell inside of a cell. And in fact, that's what we think it is. Because those ribosomes that the mitochondria uses, those are really similar to bacterial ribosomes. That DNA that's inside the mitochondria, those are really similar to bacterial DNA. So we think long ago, before eukaryotes started using oxygen, there was this bacteria that figured out how to use oxygen to make lots of energy, and one of these bigger cells kind of like ate that bacteria, but for some reason didn't destroy it. And over time, they learned that it was really convenient to live together, right? The mitochondria, or the cell that would eventually become the mitochondria, got all of its needs taken care of it, right? It's protected, it's inside this other thing. It can focus all of its time on making energy. And over time, it actually lost all of the proteins most of the proteins that aren't involved in just making ATP. Everything else is taken care of for it by the cell. And the outer cell, the eating cell, got this really efficient engine that basically, is super good at taking oxygen and making ATP. And so now mitochondria can't live on their own because they're basically hyper-specialized. They just make a ton of ATP, and they expect that everything else will be taken care of for it by the cell. Whereas the cell depends on having mitochondria to make ATP for it. They are what are called, this process is called endosymbiosis. This idea that one organism has devoured another organism, but then they learn to live in symbiotic relationship with each other and actually become so interdependent that they are now the same organism. That's pretty much all of the organelles inside of the cell. But there are, or at least all of the membrane-bound organelles inside of the cell, but there are some special features that I want to talk about as well. First off, microvilli. Now, microvilli, as you can see here, are these, microvilli are these thread-like projections coming out of the cell. And what they do is they increase the surface area of the cell, right? So why is these little thread-like things coming out of there? It increases the surface area. That means that there's something important happening on that cell's surface. A lot of what happens on the cell surface is that things are either absorbed or things are extruded. You're either taking something in or forcing something out. So anywhere in the body where you see cells that are absorbing things, for instance in your stomach, you will see a lot of these microvilli that increase surface area so that absorption can happen faster. And in cells that are supposed to release things, like for instance glandular cells that are supposed to release hormones or sweat or something like that, you'll also find microvilli there so that the cell can release things faster, because that speed is all dependent upon the amount of surface area that you have. More surface area means that it happens faster. Flagellum, which you see up here, this long thing here, and I'm going to add cilia into this, which are similar to flagella. Let's actually show you a picture here. So these are microvilli. This is a close-up picture of microvilli. And you can see that these microvilli are actually filled with these little proteins inside of them that poke them out. This finger-like microvilli border is sometimes called a brush border, because it looks like a brush, like you would brush your hair with. Flagella and cilia are motile structures. That means that they move. And cilia look a lot like microvilli, right? They're these short finger-like hairs. This is the exception that proves the rule. So remember I said whenever you see these short finger-like hairs, that's usually to increase surface area? Well, cilia are not there to increase surface area. What they are is there to move. And these are actually used by some things like paramecia to move around. But in your body, they aren't there to move cells around. They're there to move things around cells. These are moving structures, and they make this little beating motion, right? So like they stand up straight, and then they go whoosh down, like a swimmer doing a breaststroke or something like that. This is what's called the power stroke, and all of the cilia beat in unison, going whoosh, whoosh down, and that, in a single-celled organism, pushes the cell along. In your body, what it does is it moves things across the surface of the cell, particularly mucus. You have cells that secrete mucus in, say, your lungs, right? Or in your throat, your mouth, your nose. If all that mucus drains down into your lungs, that's bad. You can actually drown that way, right? Your lungs can fill up with mucus, and then you can't breathe anymore. This is what happens to people who have, like, pneumonia. So you have cells, specialized cells, in your lungs and in your trachea that basically beat their cilia to move the mucus up from your lungs, into your trachea, into your mouth, where you will, like, go and cough it out, or it will drain down into your stomach where it gets destroyed. This is called the mucociliary escalator. Basically, literally means using cilia to move mucus up. Mucociliary, cilia moving mucus, escalator. An escalator moves things up. It escalates. There are also cilia found on the cells, sensory cells in the nose. This will, like, create a wafting draft that moves air over sensory cells, and that moves mucus and fluid that the air has been dissolved in over those sensory cells so that they all get exposed to the air. They're found in uterine tubes, where they will actually kind of create a current that will catch an egg that is released from the ovaries and channel that egg down into the uterus. It's found in the ventricles of the brain. We'll talk about this more, but it actually is involved in circulating your cerebrospinal fluid around your brain and spinal cord. And they're found in the ducts of the testes, where they move sperm from your testes through a bunch of tubes inside of your testes and eventually out of your testes and into your seminal ducts, where sperm can eventually be ejaculated from. So cilia are these short thread-like projections that beat and create basically a current. They move things around cells. Flagella are really long, and you usually don't have a whole bunch of them. You might have just one or two flagella. These really long tail-like projections that do basically the same thing. They're actually structured pretty much the same. And but they're found on only really one cell in the body, and that cell is the sperm, and these ones actually are involved in moving the cell around. Once the sperm is ejaculated into the female reproductive tract, the flagella becomes activated and allows the sperm to swim towards the egg so that fertilization can take place. Last thing that I'm going to talk about is the cytoskeleton, right? Just like your body is given strength and form by your skeleton, the cell is given strength and form by the cytoskeleton. Remember, cyto means cell, so this is just literally cell skeleton. And there are three components of the cytoskeleton: microfilaments, intermediate filaments, and microtubules. Briefly, microfilaments are the smallest of them. These are made of actin protein. Actin protein is actually the protein that allows your muscles to contract. So when your muscle cells contract, that is these microfilament cytoskeleton components that are pulling the cell together. That's what they do in muscle cells. But in most cells, you will find this web of actin filaments. So see this web that's around the surface of the cell, these small things there, and they give shape to the surface of the cell. So we talked about how cells have different shapes. Well, how do you think they get those shapes? You put just like a membrane out there. That membrane wants to form a sphere. How do you make it form something that's stellate or fusiform or columnar or something like that? Well, you do it by basically making this surface structure, which is a network of these microfilaments. In addition, remember the villi, the microvilli that form the brush border? Well, the things that push out that microvilli are actin microfilaments. So those are what are responsible for the surface shape of the cell. They also can move things around inside of the cell, particularly when things need to move sideways. They move on roads made of these microfilaments. Microfilaments are strong towards compression, but like weak towards pulling. Right? If you pull a microfilament, it tends to fall apart, but if you push it, it's strong. They're like a bar. Right? Intermediate filaments, as you might guess, they're intermediate in size, and they are like ropes. All right? Ropes, you can like pull something with a rope, no problem. Ever tried to push something with a rope? Doesn't work so well. So you can pull things with it, and they can resist pulling forces really, really well. These are going to be used particularly on attachment points. So, for instance, here, this is what's called a desmosome. We'll talk about them more later on, but this is where one cell attaches to another cell. And you'll find a lot of these intermediate filaments at that desmosome, so that when you pull one cell against the other, they don't just rip and tear. They have these ropes that are keeping them connected together. You also find intermediate filaments that move, sort of throughout the cell, connecting one thing to another. Anytime you want two things to not get like torn apart, you connect them with intermediate filaments. Things don't really move down intermediate filaments, but it helps the cell resist stress and deformation. So it helps the cell resist shape-changing effects. Last, we have microtubules. These are the largest of the cytoskeletal components. Microtubules usually start towards the center of the cell. There's this organelle. It's not a membrane-bound organelle, but there's this organelle called the centrosome. And the centrosome is where microtubules are made from, and it's usually right next to the nucleus, and they go from the nucleus out towards the surface of the cell. This is actually the superhighway of the cell. They do give shape to the cell, and they hold organelles in place, but they act as a road. There are these molecules called kinesin and dynein. You can see one here, right there. There's this little guy right here, and you see it's attached. It's like walking down this microtubule, and it's attached to this vesicle. So remember how we talked about how vesicles would butt off from the endoplasmic reticulum, and then they'll go to the Golgi, and then they'll butt off from the Golgi, and they'll go to wherever their final destination is? How do they move around? They don't have a brain. They don't have a propeller or a motor system. They don't have cilia or flagella. How do they move around? They don't move around by themselves, but they get picked up by these motor proteins that then grab hold of them and walk them down cytoskeletal components. Usually microtubules go from the center of the cell out and back, and then they can get off of a microtubule and hook onto a microfilament and move side to side. And these motor proteins grab them and move them around the cells. And not just vesicles can move around this way, but organelles can as well. So for instance, mitochondria can move around the cell by scooching down microtubule pathways. These microtubules are also involved in cell division. When a cell replicates and makes another cell, these are going to be the things that push the cell apart, elongate the cell, and that pull its DNA chromosomes to opposite sides. These are the largest and strongest structures. They're very good at resisting compressive force, but again, they don't do pulling very well. They tend to fall apart if you try to pull them. But I want you to remember their function as like highways in the cell. All right, that's it for this video. Key things that you need to know: the names of all of the organelles and cell components that we talked about. You should be able to recognize and know the function of the nucleus, the smooth and rough endoplasmic reticulum, the mitochondria. I won't ask you to recognize a lysosome or peroxisome or vesicle, because they all basically look the same. But I will expect you to know the function of those three things. You should know the three types of cytoskeletal components and the main function of each. Microfilaments shape the cell surface. Intermediate filaments are like ropes. They resist stress, and they attach cells to other cells. Microtubules act as highways. They go from the center of the cell outwards. You should know the function of microvilli and what cilia are and how they are different from microvilli. So, for instance, if you have projections that move, those are cilia. If you have projections that just sort of sway whichever way the current goes and that are mostly there to increase surface area, that is microvilli. You should be able to recognize after the lab all of these components inside of a cell picture.